And so the issue became, how does DNA replication work? And so I'm about to go into it. Now, I'm going to note we're going to be starting this DNA goes to RNA, goes to protein, and DNA goes to itself, right? DNA is replicated. It makes RNA. The RNA is used to make protein. This will be what we'll be talking about today and tomorrow. So the first step of that is, how does DNA give rise to more DNA? Well, how do you find an enzyme? How do you do biochemistry? What do? Assays, so I got to grind up the cell. I got to choose a cell in which I'm likely to find an enzyme, grind it up, break it up into different fractions, and test each fraction. That's all biochemists do, right? So what cell might have the enzyme we're looking for? What cells might be able to copy DNA? How about all cells? So let's use a simple cell. What's a simple cell? Let's use bacteria. So we'll take some bacteria, we'll grow it up, we'll grind it up, we'll fractionate it into different fractions, and we'll see if one of those fractions has the ability to copy DNA. If we're going to run an assay, we have to give it a substrate. What substrate would you like to give it? What do you think it needs? It better have some free nucleotides, otherwise how are we going to make DNA? What else? You're going to ask it to make DNA all by itself? We want something that can copy one of the strands of a double helix. So what should we give it? Sorry? Half a helix, a strand of DNA. The strand to be used as a template. So let's give it a template strand. So we'll take a template strand of DNA. There's my template of DNA. Let's actually give it a little sequence actually here. Let's say A, phosphate, T, phosphate, G, phosphate, C, phosphate, A, phosphate, T, phosphate, T, phosphate, A, phosphate, G, phosphate, G, phosphate. I'm going to not write the phosphates too much longer, guys, but anyway, C phosphate, T phosphate, like that. Okay? Pretty soon, in fact, almost immediately, I'm going to start dropping the phosphates in here, but that's is the way it goes. All right? That's a template. We need floating around in the solution. Some trinucleotides. So we have some nucleotides, nucleotides floating around. And now, will this enzyme work? We're going to have an enzyme, we'll try different fractions and see if it's able to just install the right letters in the right place. Now, it turned out it needed one more thing. And the person who discovered this, Arthur Kornberg, thought of it. It needed a head start. It needed a primer. So the primer goes, let's say, phosphate T, phosphate